Hello, my name is Joanna Asper, and I work with the cell biology business at Thermo Fisher Scientific. I'm here to present this webinar about KnockOut Serum Replacement multi-species and how it enables feeder-based PSC culture, which is proven more reliable than FBS. As a brief outline, I'll start with the evolution of media systems to become more and more defined. I'll talk about where KnockOut Serum Replacement comes in that evolution, what it is, and why people use it. Ultimately, it's about better performance compared to FBS, and I'll go through what exactly those performance advantages are for both mouse and human ESCs or iPSCs, rounding off each section with how you would go about transitioning from FBS to a more defined medium with KnockOut Serum Replacement multi-species. When human ESCs were first derived by James Thomson in 1998, he used a feeder-dependent system with FBS and MEF feeder cells, which are both obviously animal origin and contribute undefined components to the culture. Since then, human PSC culture systems have evolved to become more defined. First, the introduction of KnockOut Serum Replacement allowed human PSCs to be cultured without FBS. Later, different media like StemPro, HESC, SFM allowed human PSC culture to be done in feeder-free conditions. And today we offer Essential 8 medium and Essential 8 flex medium, which are fully defined and xeno-free. Now, much of the evolution of human PSC culture media was motivated by the desire to generate clinically appropriate human PSCs, meaning a lot of the intention was to reduce the risk of contamination with adventitious agents like viruses and bacteria, and also to reduce the contribution of proteins from other animals. However, there are many benefits to more defined media systems that are valuable beyond translational applications or even beyond just human PSC work, like reduced variability of medium composition and a reduction of undefined components. Let's take the case of FBS more specifically. Fetal bovine serum is 100% animal material and contains many undefined components. Those components can vary from lot to lot, influenced by the individual differences that naturally occur between animal to animal. And those components can either be beneficial or detrimental to PSCs. That means, by detrimental, I mean it could cause differentiation or inhibit growth. And so the benefit of a more defined FBS-free medium is that it follows a set formulation that can be made more consistently, and that formulation can also specifically reduce detrimental components while keeping what is important for PSC growth. So it's clear that there are good reasons for using more defined systems, but if that's the case, then why do people still use feeder-based cultures? Well, if you look at the components of a feeder-based system, it typically contains a rich supplement like FBS or KnockOut Serum Replacement, and that provides growth factors and nutrients. And then there are also feeder cells that provide additional support. So with rich medium and extra support from feeders, PSCs are more likely to remain alive and undifferentiated despite mistakes or harsh treatments, for example, clonal expansion. And so because of that, feeder-based cultures are great for PSC researchers who are still learning new techniques, and they're also often used in parallel with feeder-free cultures as a backup. So there are reasons for using more defined systems as well as reasons for using feeder-dependent systems. In a way, KnockOut Serum Replacement offers the best of both worlds. What exactly is it? It's a more defined FBS-free culture supplement that allows researchers to enjoy the benefits of a feeder-dependent culture while using a more defined medium. It's also a well-established reagent in feeder-dependent culture, cited in more than 2,000 PSC-related publications in a variety of applications, including PSC culture, ESC derivation, reprogramming, differentiation, and so on. The next point is also very important. KnockOut Serum Replacement offers regulatory documentation to ease the transition from bench to clinic. The product was launched in 1997, which was the beginning of almost 20 years of history for the KnockOut line of products. Since then, it has been used in many seminal or landmark papers. For example, the first human somatic reprogramming paper from the Yamanaka lab. Many other knockout products have also been launched, including different basal media and other serum replacements. In 2010, KnockOut Serum Replacement became an FDA-cleared medical device in the U.S., and regulatory documentation became available in other countries as well. This year, we introduced a related product called KnockOut Serum Replacement Multi-Species. How is this different from the original KnockOut Serum Replacement? This product is for research use only and is therefore priced accordingly. It is appropriate for mouse, human, and other mammalian PSCs, but it uses the same formulation as the original product and is manufactured and qualified the same way. So you can look at the KnockOut Serum Replacement's characteristics, publication record, the 20-year history, and then you can conclude basically that KnockOut Serum Replacement Multi-Species is proven more reliable than FBS, just like the original product. You can see this in terms of a more defined formulation, a more stable supply and price, because it's not completely reliant on animal material, as well as better performance in mouse and human PSC culture, which I'll explain in the subsequent slides. Before I move on to that, just a quick reminder here that even though the focus of this presentation is on mouse and human PSCs, KnockOut Serum Replacement Multi-Species does work for a variety of mammalian PSCs, including rat, monkey, and pig. On the slide are some highly cited references using KnockOut Serum Replacement, the original formulation for different species. In later slides, I will refer to other papers that published using the original product as well, but please remember that the findings also apply to KnockOut Serum Replacement Multi-Species because they share the exact same formulation. With that out of the way, I'd like to talk about the advantages of using KnockOut Serum Replacement Multi-Species for mouse PSC culture. The first major advantage is that mouse PSCs cultured in KnockOut Serum Replacement Multi-Species form better-looking colonies compared to those cultured in FBS. As you can see in this image, colonies are more compact and dome-shaped, reminiscent of naive stem cells, and the colony edges are more defined compared to those in FBS. If you look at the gene expression of those colonies, you see bright and uniform expression of PSC markers OCT4 and SOX2. In contrast, PSCs in FBS are more heterogeneous. So that heterogeneity in serum has been confirmed in this publication from the Orkin lab in Harvard, as well as another paper referenced here. Both groups used a much more sophisticated approach, performing single-cell global gene expression analysis. As you can see in the title, they concluded that serum provokes gene expression variability, the model being that mouse ESCs grown in serum consist of a mixture of cells that are more pluripotent or primed for differentiation or already on the path to differentiation. As a result, we observe variable expression of pluripotency and differentiation genes. This is certainly a concern when performing molecular analyses, where heterogeneity of a population can potentially mask the results of the experiment. And that's not even considering the type of variability that you would expect from serum as a reagent that is 100% animal material. Notably, the Orkin lab observed more consistent gene expression in KnockOut Serum Replacement, the original formulation, compared to serum. That last slide hints that FBS is more differentiation permissive. Looking into this, we simulated a differentiation-inducing situation by culturing cells without LIF, not only comparing the effect of FBS versus KnockOut Serum Replacement Multi-Species, but also traditional DMEM versus KnockOut DMEM, which has been optimized for mouse PSCs with an osmolarity that approximates that of mouse embryonic tissue. The result is shown in the graph. This graph shows the percentage of colonies that remain undifferentiated as gauged by morphology and alkaline phosphatase staining. As you can see, colonies rapidly differentiate with FBS, although using KnockOut DMEM as a basal medium helps. As for PSCs and knockout serum replacement multi-species, they do differentiate in response to the removal of LIF, but knockout serum replacement more effectively maintains colonies in the undifferentiated state compared to FBS, synergizing well with knockout DMEM. This tells us that knockout serum replacement multi-species offers better maintenance of undifferentiated PSCs. Beyond the routine culture of mouse PSCs, the knockout serum replacement formulation also offers advantages over FBS and reprogramming. This has been reported in a number of publications, but I've only summarized data from one paper here. You can see the percentage of cells expressing GFP under the PSC-specific OCT4 promoter at day 20 of reprogramming. Zhao et al. Observed little to no GFP expression when using only FBS, or using FBS initially and then switching to knockout serum replacement later. In contrast, there was robust GFP expression in cells growing in the knockout serum replacement formulation. This actually translates to faster reprogramming and knockout serum replacement, as GFP-positive colonies emerged as early as day 14, whereas it took more than twice the amount of time, about 32 days in FBS. You may be wondering why there's such a drastic difference. According to the literature, one possible reason is that knockout serum replacement selectively supports PSCs over non-PSCs, including the parental fibroblasts. Another possible reason is decreased ERK activity when growing cells with knockout serum replacement, because ERK signaling is known to suppress mouse pluripotency. The final advantage of knockout serum replacement formulation over FBS in mouse is that it enables more efficient mouse ESC derivation. This is again shown in a number of publications, some of which are cited at the end of this deck. In this example from Davies and Fairchild, they show that epiblasts in FBS did not yield any ESCs from a non-permissive strain, while knockout serum replacement produced ESC lines from about half of the epiblasts picked. They used the same method for a number of other mouse lines and successfully generated multiple ESC lines from each one. In conclusion, knockout serum replacement is a reliable reagent for mouse ESC derivation, and it actually permits the derivation of ESCs from non-permissive lines. With all of those advantages in mind, if you were interested in switching from FBS to knockout serum replacement multi-species, it's a pretty easy transition. Mouse PSCs and FBS, whether frozen or already in culture, only need to be thawed or passaged directly into medium containing knockout serum replacement multi-species, and you only need to use standard thawing or passaging protocols. As you can see with the change in medium, you immediately see the difference in colony morphology. So that was for mouse PSCs. What about for human PSCs? We actually have very similar observations for human PSCs compared to mouse. ESCs cultured in knockout serum replacement multi-species form more compact colonies with more defined edges. You see that very clearly in the high magnification images below. If you look at the gene expression of those colonies, you see bright and uniform expression of PSC markers, SOX2 and NOT4. In contrast, PSCs and FBS are more heterogeneous, and SOX2 expression is lower as well. Other groups have observed other indications that culturing human PSCs and FBS permits differentiation, sometimes even leading to the loss of lines due to differentiation or difficulties in deriving or reprogramming lines. Replacing FBS with the knockout serum replacement formulation reduces this effect. In this particular study by Scottman et al., they did a global gene expression analysis of human ESCs grown in FBS versus knockout serum replacement. While there were many similarities in gene expression because they were both ESC cultures, there were differences as well. And clustering analysis of genes that were more than twofold different between the two conditions showed that FBS grown cells clustered more closely to fibroblasts compared to those grown in knockout serum replacement. In a subsequent RT-PCR experiment, they found that a number of pluripotency genes were expressed higher in knockout serum replacement, including OCT4, NANOG, and so on. On the other hand, genes associated with differentiation were higher in serum. Some examples are SOC17 and GATA6. This suggested that the human ESCs grown in FBS were more differentiated than those cultured in knockout serum replacement, although they morphologically appeared to be undifferentiated. As you can see in their title, their conclusion was that the gene expression signature correlated with the enhanced and prolonged growth of human ESCs in the undifferentiated stage when cultured with knockout serum replacement. As for why this might be the case, the gene expression data offers some clues as well. For example, ESCs in knockout serum replacement express higher FGFR1, a receptor for FGF2, which, as you know, plays a huge role in maintaining human PSC pluripotency. Now moving on, one advantage we see in human PSCs that we didn't see in mouse PSCs is that cells actually grow faster in knockout serum replacement multi-species compared to FBS. This is clearly shown in this proliferation curve with mean viable cell numbers over 3 passages. The last advantage I wanted to mention is that just like with mouse ESCs, knockout serum replacement improves the efficiency of human ESC derivation. Here I have data summarized from literature. In the first table, Hongmei et al. Were deriving human ESCs using 3 different batches of FBS alongside of knockout serum replacement. Only one batch of FBS supported blastocyst outgrowth, which is a really good example of that lot-to-lot variability I was talking about at the beginning of the webinar. Between that working FBS lot and the knockout serum replacement, they observed a much higher yield or efficiency of derivation in knockout serum replacement. Inzunza et al. Made a similar observation with the data table below, with improved ESC derivation using knockout serum replacement. There are a number of reasons behind this improvement. Number one, in the graph on the right, work from the Thompson lab showed that ESCs had a higher clonal efficiency in knockout serum replacement compared to FBS. Number two, again, there seems to be selective support for PSCs versus other cell types, in this case, trophectoderm. Number three, as I just showed you, PSCs grow more robustly and have a decreased propensity to differentiate in knockout serum replacement, and this could have helped the emerging PSC survive as well. With all of these advantages in mind, if you plan to transition human PSCs from FBS to replacement, that can be done pretty easily as well. Human PSCs already in culture can be directly passaged into medium. To wrap this up, let me summarize what I have said. Knockout serum replacement multi-species offers a more defined formulation for feeder-dependent culture. Through the original knockout serum replacement, it has a long history of stable price and supply, but examples include better colony morphology and better maintenance of undifferentiated PSCs, whether they are from mouse or human. Altogether, knockout serum replacement multi-species offers PSC culture that is proven more reliable than FBS.